So by binary fission, it occurs repeatedly by trophozoite stage. So type of asexual reproduction entomobiocyte, if anybody asks you, binary fission. Okay? And the same way type of nutrition or mode of nutrition, if anybody asks you, holozoic. Because they engulf the cell debris, tissue debris, RBC, bacteria, after dissolving the mucous membrane of the large intestine. So they take that. So next stage, some of the daughter entomobulae converted into daughter trophozoites converted into pre-cystic stage. So in this conversion, what happens, let us take this diagram of pre-cystic stage first of all. So in this pre-cystic stage, it loses the pseudopodia. It loses the pseudopodia. And cartwheel shaped nucleus is still left. It is remaining left in the center. Or to a side also you can draw this. This is cartwheel shaped nucleus. It is retained. Still it is retained because it is not undergoing any division. No cell division occurs in the pre-cystic stage. Pre-cystic stage generally spherical and with rod-like structures present here and there. These rod-like structures are called the ribonucleoproteins, which have reserve food material. And besides this, the glycogen granules also present. What are the reserve food materials of pre-cystic stage? 2 marks question. In the question bank, if you see, there is a 2 marks question. So, what are the reserve food materials in the pre-cystic stage? The first one is chromatoid bodies, rod-shaped structures. These are the chromatoid bodies, and these round granule-like structures are called glycogen granules. So why? Because these structures must be present in the pre-cystic stage. Feeding is stopped. There is no feeding. Trophozoite is feeding stage. This is non-feeding stage, and spherical or oval. This is the pre-cystic stage. The cartwheel nucleus also you can label. Cartwheel nucleus, and this is cytoplasm. So next to trophozoite you have to draw this diagram, pre-cystic stage, the life cycle of entamoeba is the 8 marks question. Already told you. So specifically in this structure and life cycle of entamoeba is 8 marks question. Life cycle of entamoeba only for 4 marks question. Trophozoite structure also only. So in this essay question 2 4 marks questions also there. Structure of trophozoite to life cycle of entamoeba. So like that. So these pre-cystic stages, what are the characteristics of the pre-cystic stages? Let us see one by one. Pre-cystic stage characteristics are, these are spherical or oval. Spherical or oval. Non-motile, that is trophozoite is motile. Motile means move. It moves, movement occurs. This is non-motile because no pseudopodium. It loses pseudopodium. That also you can write here. It loses pseudopodium, monopodium. Single pseudopodium is present, hence it is called monopodium. It is blunt finger-like structure, hence it is called lobopodium. That is absent in the pre-cystic stage. And non-feeding stage, non-motile, non-feeding stage. And non-pathogenic means non-harmful. It never cause any disease. So that's why during trophozoite only disease symptoms occur. But in the rest of the stages, disease symptoms never occur. So that's why it is a non-harmful stage. Pre-cystic stage is the non-harmful stage. It loses monopodium. Non-feeding stage, spherical. Contains the reserve food materials. This is 2 marks question. Reserve food material, you keep the 6th point, 2 marks question. Reserve food materials present in the pre-cystic stage in the form of rod-shaped chromatoid bodies or bars. You can say they are rod-like structures. That's why chromatoid bars or chromatoid bodies contain ribonucleoproteins. They contain ribonucleoproteins. Just like Nigel's granules. In the Nigel's granules, what type of structures present? Can any one of you remember the multipolar neuron structure? Already I told you. The cytoplasm of nerve cell body contains the Nigel's granules. Any one of the class, Varshita, Srujana, Satvika, Prabhav. No, neurofibrils are separate structures. Nigels granules, neurofibrils, they are all separate structures. Nigels granules contain RER. RER means rough endoplasmic reticulum. That's why chief site of protein synthesis in the neuron is Nigels granules. Because why? Assertion reason. Chief site of protein synthesis in the neuron cell body are Nigels granules. Reason is the Nigels granules contain RER, rough endoplasmic reticulum. In the same way, the chromatoid bars contain ribonucleoproteins, and another second reserve food materials are glycogen granules. Glycogen is converted into glucose and taken away. This is about the pre-cystic stage. Then afterwards, what is that stage? Third stage in the life cycle, cystic stage. The chitinous cyst is formed around the pre-cystic stage. So that's why it is called cystic stage. So first point, around the pre-cystic stage, of course, in the book it is not given, the cyst made up of chitin, a material chitin with which the cyst is made up of. So around the pre-cystic stage, the cyst formation occurs. Pre-cystic stage, where it occurs, pre-cystic stage occur in the, that also you can write down. Both pre-cystic and cystic stages are occur in the lumen of the gut. Lumen of the gut means large intestine. In the lumen cavity. That is why it is pre-cystic stage and cystic stages are Cenozoic. Around the pre-cystic stage, what happens around the pre-cystic stage? The cyst made up of chitin is formed. Cyst made up of chitin is formed. Okay, so that is cyst made up of chitin. That's why it is a beautiful protection of cysts when they come out of the feces of the humans. Generally, along with feces, the cysts come out, and they stay for the period of 10 days in the moist soil. When houseflies and cockroaches, mechanical vectors come, definitely they contaminate our food and drinking water. Along with food and drinking water, they enter into our human gut again. So thus, around the pre-cystic stage, cyst made up of chitin is formed. So this process is known as encystation. This process is known as encystation. So encystation is a best process and phenomenon in which the cysts are being protected outside of the human host body. Outside the body of the human host, they are released. So there they are being protected by this cyst made up of chitin. Unfavorable conditions are tied out. To tide over the unfavorable conditions, this encystation phenomena occurs. Now let us go to the next page. In the cystic stage, the second point is it also contains chromatoid. Continuously they are present. It also contains chromatoid bodies or bars, you can say, whatever it is, and glycogen granules as reserve food. That is continued up to cystic stage. Because these are non-feeding stages, they have to survive depending on this reserve food, both pre-cystic and cystic stages. The nucleus undergoes two mitotic divisions. Now this is called cystic stage. Nucleus of pre-cystic stage undergoes two mitotic divisions. Mention the number. First mitotic divisions, how many nuclei are formed? 2. Second mitotic division, another 2 added. Means 1 becomes 2, 2 becomes 4. So a nucleus undergoes two mitotic divisions to form 4 daughter nuclei. 4 daughter nuclei. This point is very important as far as cystic stage is concerned. So this cyst is described as or is known as tetranucleated cyst. Tetra means 4. Nucleated means nucleate. Tetranucleated cyst or put a slash quadranucleated cyst. Tetra or quadranucleated. So all these alternative terms may be given or may not be given in the book, but you have to take down. Tetranucleated cyst or quadranucleated cyst. So this is the infective stage of Entamoeba histolytica, which is an infective stage of Entamoeba histolytica to man. At this stage only the parasite enter into new host, so which is an infective stage to man. At what stage the Entamoeba enters into man? Tetranucleated cyst. So what is the infective stage of Entamoeba histolytica to man? If it is asked, your answer must be tetranucleated cyst or quadranucleated cyst. Very, very important point. This point is very, very important point. Remember this point. Now, so these cysts enter into the by contaminated food. By contamination of food by houseflies and vectors. By contamination of food and water. By mechanical vectors. What are the types? How many vectors we have given? 2 vectors. 2 types, mechanical and biological vectors. The vector in which the parasite never undergo any development is called mechanical vector, but carries only disease, infective stages of the parasites. So after food and water contamination by mechanical vectors, say in the bracket housefly and cockroach. Houseflies and cockroaches are the mechanical vectors for the Entamoeba histolytica and Ascaris lumbricoides, who are the monogenetic parasites. So whenever the monogenetic parasites means the parasites which complete their life cycle in only one host, single host, that is monogenetic parasites. So by contamination of food and drinking water by mechanical vectors, the cysts enter into the quadranucleated cyst. Simply says cysts enter into the gut. Cysts enter into the gut of the man. Now let us take this tetranucleated cyst. This is tetranucleated cyst. This is cyst wall, and now actual Entamoeba histolytica is this one, and then the nuclei is divided into 4 daughter nuclei. These are the 4 daughter nuclei with chromatoid bars and glycogen granules here and there. So this is cyst. These are nuclei. So this is the cystic stage. This diagram, the cystic stage also completed description. So by contamination they enter into our human gut. So now the metacystic stage will be formed. This is the 4th stage in the life cycle of Entamoeba histolytica. This stage is called metacystic stage. How the metacysts are formed? Let us see. When the cysts enter into the gut, infective stages, these are infective stages. That's why when the cysts enter into the gut of person or a new host is man, gut of new host, the cyst is dissolved by the enzyme trypsin. It is a pancreatic enzyme, you know, by the enzyme trypsin. In the small intestine only it is dissolved. In the small intestine only it is dissolved. Cyst is dissolved. So now the Entamoeba is free. When the cyst is dissolved by the enzyme trypsin, cyst is dissolved. Now this process is called, you say like that, this process means removal of cyst or digestion of cyst by trypsin enzyme, is called excistation. Excistation. So those amoeba are called excistic amoeba. Means amoeba without entamoeba without cyst are called excistic amoeba. Amoeba are also called metacysts. They are also called metacysts. Other name of after excistation, the cystic stage is called metacyst. So in this metacyst, the third mitotic division occurs. In the metacyst, third mitotic division occurs to produce how many daughter nuclei? There are already in the infective stage, cystic stage, there are already 4 daughter nuclei are there. That's why it is tetranucleated cyst. Right here, tetranucleated cyst. That is why it is so called. So, infective stage. These 4 nuclei undergo third mitotic division to produce 8 daughter nuclei. So 4 becomes 8. Like this, you have to complete the finish. Today it is completed. So in the metacyst, third mitotic division occurs to produce 8 daughter nuclei. Each nucleus of which each nucleus is surrounded by a bit of cytoplasm. Each nucleus is surrounded by a bit of cytoplasm. So how many daughter nuclei are there? 8. Cytoplasm also divided into 8 bits. So each nucleus is surrounded by a bit of cytoplasm. Now these stages are called metacystic trophozoites. These stages are called metacystic trophozoites. So you say like that. These are called metacystic trophozoites. Gradually they will be converted into trophozoites and settled in the mucosa of large intestine and again undergo binary fission to produce into many daughter entomobulae, and some of the daughter entomobulae converted into precystic stage losing pseudopodium, and precystic entered into cystic, and cystic after existation in the small intestine they will be converted into metacysts. The metacyst third mitotic division occurs to produce 8 daughter nuclei. Each nucleus bit is surrounded by a bit of cytoplasm. Now these daughter cells are called metacystic trophozoites. So encystation occurs here like this. This is encystation. In this, the daughter nuclei are how many? 8 daughter nuclei are there. These are all 8 daughter nuclei. All these are 8 daughter nuclei. So each daughter nucleus, now in the second step, each daughter nucleus is surrounded by this is each daughter nucleus. How many? 8 daughter nuclei are there. Means how many metacystic trophozoites are developed? They are also 8. So each daughter nucleus is surrounded by a bit of cytoplasm. Like this. Now this metacyst is broken to release these trophozoites. So these are the this is metacyst. This is metacyst. These are metacystic trophozoites. 8 daughter entomobulae are produced. These daughter entomobulae are called metacystic trophozoites. That is why one cyst if it enters in the human body, 8 daughter entomobulae are produced. It's very dangerous. So how many cysts we are taking along with water and food, contaminated food. That's why open utensils the food you should not take. Crowded places where the breakfast and these things should not junk food. If you go and take junk food in the crowded areas, generally Pani Puri, all these areas, definitely these cysts are present. You don't know who is suffering from amoebiasis. So that's why they contaminate. Like that, even houseflies and cockroaches also bring the cysts and drop on the utensils, containers, food containers and water containers without lids. They are dropped into that. So contaminated. Thus it is enters into. So this is the life cycle is completed. Okay. And then pathogenicity. This is also another 4 marks question. Life cycle 1 4 marks question, trophozoite 1 4 marks question, pathogenicity also another 4 marks question. You see how important it is. I need not separately say that it is very important. You read carefully, go through carefully. Should I? Should I say? No, not at all necessary. So that's why you must study the entire life cycle. Pathogenicity means disease, how the so the disease caused by this parasite is called amoebiasis. Amoebiasis are also called intestinal amoebiasis. This amoebiasis is intestinal amoebiasis, or they can also say amoebic dysentery. Amoebic dysentery. This is the first pathogenic symptoms. And secondly, symptom is amoebic dysentery symptom is blood and mucous motions. Blood and mucous motions. Because during the mucous membrane of large intestine is dissolved by the enzyme histolysin, the ulcers are broken, abscesses are broken. The abscess from which the mucus comes out. So thus along with blood mucus also comes out. This is the best symptom. And sometimes asymptomatic cyst passers are there. Asymptomatic means no blood and mucous motions. These asymptomatic people carry the cysts. Asymptomatic people carry cysts. So that can spread to new hosts. That can easily spread to new hosts. Easily spread to new hosts. So with blue ink I am writing this pathogenicity because this is a separate 4 marks question. So with pathogenicity you will complete the 8 marks essay question. Asymptomatic people, carriers, asymptomatic carriers carriers you say, carry cysts that can easily spread to new hosts. Along with their feces they come out. But the symptoms do not appear in such people. Some are asymptomatic because of immune system and all these things. And some are not prone to, not prone to appear the symptoms. They are asymptomatic. Some are prone to appear the symptoms. So like that differences are there from person to person. And then sometimes the amoebiasis, fourth heading under the pathogenicity, it is also spread. It is also spread to the other parts of the body through blood circulation. Most important point, very dangerous than intestinal amoebiasis, through blood circulation. They reach the other parts of the body. How you know? Other parts of the body, for example, first this is called other parts of the body when it reaches. This is known as through blood circulation, known as, you write down that, extra intestinal amoebiasis. The previous one is intestinal amoebiasis. This is extra intestinal amoebiasis because outside the intestine they go to the other parts of the body like liver, heart, lungs, brain, kidneys, gonads, etc. So mainly other parts of the body infected by, mainly the other parts of the body are main liver, or liver. From intestine through hepatic portal veins they reach the liver, or liver, heart, liver to heart through postcaval vein, heart to lungs through pulmonary arteries, and lungs to from any other area, brain, kidneys, even gonads also are affected. Gonads, these are affected. So other parts carefully remember: liver, heart, lungs, brain, kidney and gonads. Main liver. If liver is affected, that is called hepatic amebiasis. Very dangerous and more dangerous. Hepatic amebiasis. All are extra, come under the extraintestinal amebiasis, but this is especially liver, very dangerous. Hepatic amebiasis, the person will collapse immediately. So in the chronic patients, it is uncurable. In the chronic patients, it is not curable. Now and then it appears. That's why not curable. So this is about the pathogenicity. And prophylaxis means control. How the drugs are there, but this is most important. First control is personal hygiene. You must maintain personal hygiene. And septic toilets must be used. Septic, last small paragraph is there on the below the diagram. You see that below the life cycle diagram it is given. Septic toilets must not open toilets. Don't go to the open toilets. Personal hygiene is required. Septic toilets are required. And washing of vegetables before washing of vegetables before they are cooked, and thoroughly cooked. They must be thoroughly cooked so that cysts will die. Also, food is thoroughly cooked. If you take these precautionary measures and don't prick the nails with the mouth, nail cutters must be used every 15 days or once in a week or once in a 15 days. You can remove the nails, tips of the nails, because in the crevices of the nails also the cysts may be they are present. That's why to avoid that you have to take these precautionary measures. This is about the Entamoeba 8 marks question. In this 3-4 marks questions also are there. So the next parasite, second protozoan parasite is Plasmodium vivax. This is the full form. Plasmodium vivax. This Plasmodium vivax is commonly known as introduction. I am giving you better you note down introduction about the Plasmodium vivax. So today we will complete only introduction. Tomorrow we start the life cycle. And introduction. This is commonly known as malarial parasite. Commonly called malarial parasite. First point. And systematic position of Plasmodium vivax also let us take here. Phylum Protozoa. Right top corner you write down the classification always. And subphylum Epicomplexa or Sporozoa. Epicomplexa is the other name of Sporozoa. Write down in the bracket Sporozoa also you can write down. Phylum Protozoa, subphylum Epicomplexa. In the bracket you write down Sporozoa. And class Telosporia. Telosporia. Genus Plasmodium and species is Vivax. You need not write down. You know that here it is pretty well. Plasmodium genus and Vivax is the species. So malarial parasite and pathogenic, more harmful, because this is a monogenetic parasite. Entamoeba histolytica is monogenetic protozoan parasite. Monogenetic protozoan parasite, but this is digenetic protozoan parasite. So digenetic completes its life cycle in 2 hosts. Digenetic parasite The main host, female Anopheles mosquito, carefully write down this. Main host is female Anopheles mosquito. Female Anopheles, you simply say Anopheles means mosquito. And the secondary host, primary host or main host is that one, secondary host is man. That's why it is a digenetic parasite. It is cytozoic parasite because it lives in the RBC and liver cells. So both are cells, liver cells and red blood cells, both are cells. In these cells it is living, that's why it is cytozoic parasite. Digenetic cytozoic pathogenic protozoan parasite is Plasmodium. Okay, monogenetic histozoic pathogenic parasite, protozoan parasite is Entamoeba. Two protozoan parasites in the protozoa. One is Entamoeba, we have completed, and this is Plasmodium vivax, cytozoic parasites. You know, in the mosquitoes, they are extracellular. Cytozoic means intracellular. In man only, you write down like that, intracellular in man, but in the mosquito it is extracellular, outside the cells, means in the stomach, in the crop they live, or outside the crop they live. That's why extracellular in the mosquito. So that's why it is both extracellular and intracellular parasite. These are all introductory features of the Plasmodium vivax, and there are 4 types of species of Plasmodium. 4 types of species. Of course, all the 4 types of species are not given in the NCERT. Only 2 types of species only given in the NCERT. Anyhow, let us take those 4 types of species with the malaria fever caused by those species. First, Plasmodium vivax. Plasmodium vivax causes the fever benign tertian malaria. Benign tertian malaria fever is caused by this vivax. What is benign tertian? Every 48 hours the fever recurs. Maximum all the 4 parasites, 3 parasites in which the fever recurs every 48 hours. That's why the name tertian. So if there is no tertian, suppose the tertian is not added, then it is not every 48 hours. More than 48 hours means every 72 hours the fever comes. So Plasmodium vivax, Plasmodium falciparum, the second one, second species, more dangerous malarial fever, harmful malarial parasite, causes malignant tertian malaria. Malignant in the name itself, it is there, bad. Malignant tertian malaria is caused by the Plasmodium falciparum. And Plasmodium ovale, mild tertian malaria. Plasmodium third species, ovale. It causes mild tertian fever, not that much severity. It shows mild tertian. So all these 3 have tertian suffixed by tertian. Tertian means every 48 hours the fever recurs. This is not given in the any book. You note down this point. Why the tertian is named benign tertian, malignant tertian, mild tertian? Why it is given? Every 48 hours it recurs. But in this case, every 72 hours. That's why we should not say tertian malaria. This is non-tertian malaria. Plasmodium malariae, 4th species. This causes the quartan malaria. You should not say quartan tertian malaria. It is quartan malaria. Quartan malaria is caused by malaria because every 72 hours the fever recovers, every 4th day or every 72 hours. All these 3 every 48 hours. These are the 4 types of species of Plasmodium vivax. But we are giving detailed explanation on Plasmodium vivax only. That's why the heading is Plasmodium vivax. The most common, next point, the most common species, also bit. The most common is Plasmodium vivax. And rarest species, rarely found, is Plasmodium ovale, is the rarest species. These two are also very important. And harmful malaria fever is caused by more harmful malaria, deadly dangerous malaria or harmful malaria and tropical malaria. So many names are there. Tropical malaria, also cerebral malaria, brain also damages with this cerebral malaria. These names are there, caused by Plasmodium falciparum. Very dangerous. Malignant tertian malaria is very dangerous malaria. It leads to death also. It is lethal to man. Lethal means prananastam. So it is harmful. Our life is carried out. So dangerous. These are the introductory points. 10 introductory points are there regarding Plasmodium vivax, including classification, systematic position, phylum Protozoa, subphylum Epicomplexa, class Tylosporia. Then history of malaria. Let us take this. History of malaria, in which the Italian language malaria term is a derivative of Italian language. This malaria term malaria means bad air. Mal means bad, area means air. But it is not, its meaning is correct. But the name given to disease malaria is wrong actually. Because never the malaria fever spreads by air. It spreads by mosquito bite. It is an inoculative parasite. Mosquito injects the sporozoid stages into the blood of the human beings. That injection of the sporozoids into the blood of the man by mosquito bite is called inoculation. So that's why it's an inoculative parasite. Italian language malaria, a bad air. And secondly, Charles Lavran, a French doctor, French army doctor is there, Charles Lavran, found malaria in RBC of man for the first time. Found in RBC of man for the first time. So in the history there are scientists are there. First scientist Lavran, father of malariology also Lavran. Study of malaria, malariology, and father of malariology also Lavran. Let us go to the next page. Third scientist is a Scottish doctor, Patrick Manson. Is a Scottish doctor, Scotland doctor or a Scottish doctor. He said that malaria spread by mosquitoes. Malaria, not only malaria, while working in the filariasis, during filariasis, malaria and both malaria and filaria spread by mosquitoes. Said by whom? Spread by mosquitoes. Said by Patrick Manson. Very important point. Patrick Manson, Scottish doctor, said that malaria, filaria both spread by mosquitoes from one host to another host. And then Ronald Ross, next scientist. He is a British Army doctor in Indian Army in Andhra Pradesh, Secunderabad. While he was working as doctor in the Secunderabad Army, a British Army doctor. In 1902, Nobel Prize he has received. In 1902, he found the oocysts. He found the oocysts. On the stomach wall it is given, but it is not correct. Oocysts on the wall of the crop. On the wall of the crop. So almost introduction is completed, but in some it is written stomach. So in the bracket you keep stomach. And last and final, that's why in August, on August 20th. So August 20th is declared as World Mosquito Day. But World Malaria Day also declared as World Mosquito Day and Malaria Day, because in the mosquito stomach he has found these. But April 2007, April 20, 25th April I think, 2007, after 2007 declared as Malaria Day by WHO. So Malaria Day specially declared by WHO. WHO after 2007. This is in the 1902 Mosquito Day. About the last and final one is Grassi described the life cycle of Plasmodium in mosquito. Total life cycle described by this scientist Grassi in mosquito. This is the history of malarial parasite. So I can stop. Already we have started today Plasmodium also. More introductory features we have given. Tomorrow we can start the 2 8 marks questions live. Okay, I can stop. Bye. Okay, ma. Thank you. Thank you.